Good morning, dears. Welcome you to the next session of zoology. Today we are dealing with another disease, that is cancer. You all know that cancer is a very dreadful disease prevailing in our human community, isn't it? And it is a major cause of death too. When we are taking consideration our Indian population, large number of people are suffering from, millions of people are suffering from this dreadful disease, cancer, and large number of people die from them annually. And the underlying principle or the underlying mechanism which prevailing what causes cancer, their growth, their development, their proliferation, etc., and their treatment, control, etc., are some of the subjects which have more intensity in the field of researchers in biology and also in medicine. Day to day, very vast studies are undertaken in this subject, cancer, how we can treat them with much more effective drugs or other parameters. Okay, so in this topic, we will be dealing with what are cancer cells, how it is being diagnosed, how they are treated. Okay, so you all know that in our body, our normal body cells are growing, isn't it? The growth of normal body cells are controlled in a very particular fashion. They are controlled and regulated. Clear? So then the normal cells division and proliferations are in a controlled mechanism pattern. But in the case of cancer cell, there is a breakup in this regulatory mechanism. That is why without any control, they are dividing and dividing. Clear? So the normal cell along with this exhibit a unique property. That unique property is known as contact inhibition property. What do you mean by contact inhibition property? That means the contact of a cell with another cell in an organ will inhibit the uncontrolled growth of cell. Clear? The contact of the cell with another cell will inhibit the uncontrolled growth of the cells. But in cancerous cells, they lack this property. That is why they are dividing rapidly and intensely. Okay, so due to the rapid division of the cancer cell, they would transform to tumor cells. Okay. So these tumor cells are of two types. They include benign tumor and malignant tumor. Two types of tumor cells are there, benign tumor and malignant tumor. When we are taking consideration this benign tumor, they are confined to the location itself. Where they are originated, they will be confined to that location itself. That means they are not spreading from one place to another. Clear? Okay. So when we are comparing benign and malignant, benign tumors are less serious and they cause less damage to the body, whereas malignant tumor, they are a mass of proliferating cells, and that cells are known as neoplastic cells or tumor cells. Clear? Neoplastic cells or tumor cells. Unlike benign tumor, malignant tumor spread from one place to another place, and they also compete with the normal surrounding cells for the vital nutrients, and they will succeed in that competition and the normal cells would starve, and that would lead to the death of normal cell, and they would invade and divide rapidly, and the cells are flowed through or distributed through the blood, and wherever they are lodged, they would start to create the tumor cells in that particular area from the actual or original location. They will be transported with the help of blood to different parts of the body. But this property of transport of these tumor cells, and wherever they are lodged, they would transform into tumor cells. That this property of malignant tumors are known as metastasis. What it is known as? Metastasis. This is the one of the most feared property of malignant tumor. This malignant tumor, when it is unnoticed or when it is left untreated, it would lead to death. Or if and all it is treated, it is a lethal tumor condition. Okay, so two types of tumors are the malignant tumors and benign tumor. You have to differentiate it and study it. Okay, so that's all about an introduction about the cancer. Now we are going to see the next version, causes of cancer. Causes of cancer. The transformation of normal cell to cancerous cell are induced by some agents, and that agents are known as carcinogens. Carcinogens are the agents which induces the transformation of normal cell to cancerous cell. Okay, this agent can be physical, chemical, and even biological. Clear? The agents can be either physical or chemical or biological. Okay. The physical agents includes the ionizing and non-ionizing radiation. Ionizing and non-ionizing radiation. Ionizing radiation includes X-rays, gamma rays. Non-ionizing radiation includes UV rays, ultraviolet rays. All these rays can cause a serious damage to the DNA of our normal cell, that causes the transformation of the normal cell into the cancerous cell. Clear? And the chemical agents includes the tobacco that is present in cigarettes that we are using for smoking. You know that smoking is a serious issue for the lung cancer. Again, biological agents include certain virus. The virus which causes cancer are known as oncogenic virus. They contain a gene known as oncogenes, which are responsible for the transformation to normal cell to the cancerous cell. Along with that, our normal cell is also having some of the normal cell is found to have some gene. That gene is known as cellular gene, or otherwise known as protogenes. Clear? Cellular genes or protogenes. These genes have a property to transform the normal cell to cancerous cell under certain condition. Under certain condition, this gene will transform to oncogenes. That's why we are advised not to expose to the X-rays or the UV rays regularly. In case of X-rays and all, they have the capability to change the normal genes to cancerous gene, oncogenes. Clear? So that's all about the causes of what? Cancer, which include physical, chemical and biological agents, that is typically termed as carcinogens, and we discuss in detail each one. Okay, so next we are moving to the another topic. So we discussed what is cancer and its cause. Now we are going to see the how cancer is diagnosed or how cancer is detected. Detection and diagnosis. Detection and diagnosis of cancer. So you all know that when we detect cancer earlier, we could help the patient out from the death, isn't it? Some treatment would help to rescue that patient from the death. When we detect the cancer in the earlier stages, you might have heard that doctors are telling, yes, we could treat it and rescue the patient since the stage is 1 or 2. And sometimes they tell that we are helpless because the stage has come to 4th stage. Now we can't do anything, we can't help it out. So from telling this, we could say that earlier detection help from this help it out condition. Okay. So how we can detect this? Cancer detection is done by biopsy, right? Biopsy and also certain histopathological studies of tissues, cells, blood and bone marrow. And in histopathological studies, what it is done, cells are count. You know that cancerous cells are proliferating, dividing cells. So in these studies, the cells will be counted to check whether they have been increased or decreased, and they are done by special pathologists. And this cell counts or histopathological studies and all are done for leukemia. For what? Leukemia. Okay. So in the case of biology, the suspected tissue will be collected. One of the pieces of the suspected tissue will be collected and it will be cut into thin small pieces and they will be stained and are observed under the microscope. Okay, this is done in the case of biopsy, and this observation under microscope is done by a pathologist. Another thing, we are using some techniques, some techniques which includes radiography, that is using of X-rays, CT, MRI. You have to study the expanded form of CT and MRI, both abbreviation and also the expanded form. CT is computed tomography and it is abbreviated to CT. MRI is magnetic resonance imaging. Clear? In short, it is known as MRI. Okay. So in radiography, X-rays are used. Along with that, in computed tomography also, they utilizes the X-rays which help in a view of a 3-dimensional view of the image that is being observed or the object. The object will be reflected as a 3D image in computer tomography with the help of X-rays. Clear? In magnetic resonance imaging, as the name suggests, it utilizes the magnetic field. Along with that, some ionizing radiations are also used to get the clear picture, and in magnetic resonance imaging, it is able to locate the pathological and physiological changes in the living tissue. So, it is more effective. Okay. So, along with this, certain techniques such as antibodies are also used. Antibodies which are capable to detect certain antigen specific cancerous cells. They are being used. Such antibodies are also used, and also some of the techniques have been developed in molecular biology in order to detect and diagnose cancer. Clear? So, these are the typical things or techniques that is being used to detect the and diagnose the cancerous cell. Okay, now we are moving to the last session of cancer. What is that? Yes, it is the treatment. Now we have detected the cancer cell. Now we have to treat them. So we are moving to that area, treatment. You all know that the common treatment advised for cancer is obviously surgery. Along with that, we use radiation therapy and also some chemotherapy, and chemotherapeutic drugs are also used. Clear? So, in the what is done in radiation? In the case of radiation, radiating rays will be used to damage the tumorous cell, but extra care has to be taken to note that only the tumor cells are being destroyed with this radiation, and meanwhile along with that the normal cell should be protected from these heavy radiations. Clear? And also chemotherapeutic drugs are also given. All these are having its own, this radiation and chemotherapeutic drugs and all are having its own side effects. You might have seen the cancer prone patients and all, hair might be lost, they will be so lean and all. So that is a side effect of these things and all. After applying these things, weight loss will be the loss of appetite will be the hair loss will be the. May Almighty help us from not having these dreadful diseases, all of us from this dreadful disease. We can pray for that. Clear? And generally altogether this is being done in a combined manner. Surgery, after surgery this radiation and chemotherapeutic drugs would be given to the patients. Okay. So in some cases, in some patients are given biological response modifiers, and they include in giving alpha interferons. Some of the patients will be given some biological response modifier. Typically, it is alpha interferons. They would boost up the immune system of that patient. So, today we discussed only a small topic, cancer. What is cancer? Types of cancer, then causes of cancer, its diagnosis, detection and diagnosis, and also the treatment. I hope today's class is clear. Read your textbook clearly, and if you are finding any doubt, please ask me. Okay, thank you.